Welcome students to this lecture which will deal with the histology of the stomach and the intestines. First we'll start with the histology of the stomach. Now, the wall of the stomach has the same general layout seen in the rest of the alimentary tract. Namely, the wall of the stomach is formed of mucosa. This is the mucosa, the layer closest to the lumen, and it is formed of epithelium, underlying connective tissue, which is called lamina propria, and here we have muscularis mucosae, the layer of smooth muscle cells that separates the mucosa from the second layer, which is the connective tissue submucosa. Then, deep to it, we have the muscularis externa, and the outermost layer of the wall of the stomach is serosa. So these are the parts, these are the layers of the wall of the stomach, similar to those found in the rest of the alimentary tract. Of course, there are characteristic features of the stomach which we are going to talk about throughout this lecture. Now, first of all, if we look at the inner surface of an empty stomach, we'll see that we have several longitudinal folds. These folds that pass parallel to the longitudinal axis of the stomach. Now here, this is the direction of the longitudinal axis of the stomach. Notice how the folds are run in the same direction. Now, if we examine these folds, we will see that they are formed of mucosa and submucosa. So these are folds of mucosa and submucosa, and these folds are called rugae. These folds of the stomach are called rugae. Now we said that we find these folds on the inner surface of an empty stomach because when the stomach is full, when the stomach increases in size, these folds will disappear. And actually, this is a function of these folds to allow the stomach to be distended, to allow it to increase in size. So let's start with the first layer of the stomach wall, which is the mucosa. And as we have said, the mucosa is formed of epithelium, underlying connective tissue lamina propria, and muscularis mucosae. So we start with the epithelium. The stomach is lined by simple columnar epithelium, and this simple columnar epithelium begins suddenly at the gastroesophageal junction. At the junction between the esophagus and the stomach, if we look at this image here, this is the esophagus, this is the esophagus, this is the epithelium of the esophagus. And as you know, the epithelium of the esophagus is stratified squamous non-keratinized epithelium. Here we have the stomach. More precisely, this is the cardia of the stomach, the first part of the stomach, where the esophagus meets the stomach, the cardia. Notice the epithelium. It is a very thin epithelium because it is simple columnar epithelium. And look at the transition between these two types of epithelium. It happens suddenly. So at the site where the esophagus meets the stomach, the stratified squamous epithelium will stop suddenly and the simple columnar epithelium will begin. Here we can also see, this is a diagram that shows the site of the junction between the esophagus and the stomach. This is the esophagus and this is the stomach. Now, as you can see, there are some differences, gross differences between the mucosa of the esophagus and the mucosa of the stomach. If we look at the mucosa of the esophagus, we'll see that it is different from the mucosa of the stomach. So what are these differences? This is very important when we do endoscopy. When an endoscope is passed through the esophagus and from the esophagus into the stomach, how can we differentiate between the mucosa of these two organs? How do we know that we have left the esophagus and entered the stomach? There are certain differences between the appearance of the esophageal and gastric mucosae as seen by the endoscope. The esophagus has pink color mucosa, whereas the mucosa of the stomach is red. The mucosa of the stomach is red. The mucosa of the esophagus is not shiny, whereas the mucosa is shiny. Why is it shiny? Due to the presence of the mucus layer. The mucus layer. We are going to talk about that in a little bit. Also, if we look at the surface of the esophagus, we see that it is smooth. It has no folds, whereas in the stomach there are folds, which are the rugae that we have just talked about. Now, of course, sometimes when we do endoscopy, we inflate the stomach, and if we inflate the stomach, the folds will disappear. But usually, in the usual case, if we do not inflate it, of course, we will see the folds. And in this image, notice this is the esophagus. Here we have the esophagus. The mucosa is pinkish. It is not shiny, and there are no folds. In the stomach, notice it is folded. It has folds. These are the rugae. And you can see it is shiny. These are all droplets of mucosa, of mucus. These are all mucus. And notice the color, it is red. The junction between them, this irregular line here, this is the gastroesophageal junction. Now, if we examine the epithelium, we will see that the epithelium of the stomach invaginates the lamina propria to form gastric pits. So, and this is scanning electron microscope image. This is the epithelium of the stomach, and it passes down, invaginates into the lamina propria. Again, here we have, this is the epithelium, and it passes down into the lamina propria to form what we call gastric pits. And into these pits, branch tubular gastric glands open. So it is branched, which means that there are several secretory portions that open into the gastric pits. So the gastric pits are considered as ducts to the gastric glands. And several of these glands open into this one gastric pit. And it is tubular in shape, tube-like. The secretory portions are tube-like in shape. And usually these gastric glands are divided into regions. The isthmus, which is at the site of junction with the pit. The isthmus is the part of the gastric glands that meets the gastric pit. After that, we have the neck, a constricted region in the gland, and finally the base, the deepest and widest part of the gland. It reaches down deep into the lamina propria. Now, what are the cells that form the gastric epithelium? Many types of cells are found in the epithelium of the stomach. First, we have surface mucus cell, neck mucus cells. So as you can see here, we have two types of mucus cells. One of them is called surface and the other is called neck. Other cells are the parietal cells, chief cells, G cells, which is a type of endocrine cells, and finally stem cells. We will talk about the features of each of these types of cells and where do we usually find them. Now the surface mucus cells. These cells line the lumen of the stomach and the gastric pits. The cells are columnar in shape, and these are the cells that make up the simple columnar epithelium. The basal part of the cell contains the nucleus, which is oval in shape and rough endoplasmic reticulum, whereas the apical part contains several mucinogene granules. These are surface mucus cells, so their function is the production of mucus. The mucus secreted by these cells, first it forms a thick, viscous, gel-like coat on the surface of the stomach that can be easily seen. And this is why we said when we look at the mucosa of the stomach through an endoscope, we see it shiny because it is covered by this mucus coat. This coat acts as a physical barrier against microorganisms and the abrasive effects of foods in the stomach. Also, this mucus contains bicarbonate ions, and this neutralizes the acid in the lumen, thus protecting the wall of the stomach. Prostaglandins increases the thickness of the mucus and the contents of the bicarbonate ions in the mucus produced by these cells. And thus, prostaglandins enhances the protective properties of these cells. Aspirin and other non-steroidal anti-inflammatory drugs reduce the production of prostaglandins. So these drugs, aspirin and the other NSAIDs, reduce the protection provided by the surface mucus cells. Neck mucus cells are found in the neck region of the gastric glands, and they produce thin mucus that does not form a visible coat over the mucosa. So the surface and neck mucus cells differ in location and in the type of mucus produced. Okay, so it's not just the location, but also what type of mucus produced by the cell. Stem cells are, from their name, undifferentiated highly mitotic cells. They are usually present in the neck region of the gastric glands. And these stem cells divide and differentiate to form the surface epithelial cells and all the other cells of the gastric glands. Chief cells are usually found in the lower part of the gland. They have abundant rough endoplasmic reticulum in the basal region. In the basal region of the cell, we have rough endoplasmic reticulum, which makes this part basophilic. On the other hand, the apical region of the cell contains acidophilic secretory vesicle. Now, what do these vesicles contain? What is the function of the chief cells? The secretion of pepsinogen and gastric lipase. So these cells, chief cells, are protein-producing cells. This is why we have abundant of rough endoplasmic reticulum, and this is why the granules, the secretory granules or the secretory vesicles are acidophilic. And they are found in the apical region of the cell because the content of these vesicles are secreted into the lumen of the stomach. Then we have parietal cells, also called oxyntic cells. This is a unique cell in the stomach, and it has features that are not found in any other cell in the body. Parietal cells are found in the upper part of the gland, of the gastric glands. They are large pyramidal cells. They are large in size and they are pyramidal in shape. They have a centrally located nucleus with abundant mitochondria. Because they have the cytoplasm contains an abundance of mitochondria, the cytoplasm is eosinophilic or acidophilic. So usually they appear red in color. Now, parietal cells have some special features, again, not found in any other cell in the body, and these features depend on the activity of the cell, which we are going to talk about in the next slide. Also, it has a long lifespan, about 200 days. Most of the cells of the gastric epithelium and the gastric glands have shorter lifespan. However, parietal cells have a relatively long lifespan of about 200 days. What are the functions of parietal cells? Secretion of hydrochloric acid. The acid secreted into the lumen of the stomach, which is essential for digestion, is secreted by parietal cells. Also, parietal cells secretes intrinsic factor. Intrinsic factor is an important factor for the absorption of vitamin B12. Intrinsic factor is important for the absorption of vitamin B12, and vitamin B12 is important for erythropoiesis, for the formation of red blood cells. So what are these features? What are these special features of parietal cells that depend on the activity? Here we have a composite image. The image is of a parietal cells and it forms of two halves. On the right side, we have the parietal cell when it is active. On the left side, we have parietal cells when it is resting or when it is inactive. In the active phase, the cell has a deep circular invagination, which is called the intracellular canalicus. Notice here, here we have a deep invagination that, notice where it reaches very deep into the cell. This is the intracellular canalicus. Into this canalicus protrude numerous microvilli. As you can see, the MV, all these are microvilli. All these are microvilli that protrude, that projects into the intracellular canalicus. Of course, the microvilli increases the surface area for secretion. When the cell is resting, when it is inactive, the intracellular canalicus is short. It is not deep. Notice here, this is the intracellular canalicus. It is not deep, and the microvilli are absent. However, the cytoplasm is filled with several tubular vesicular structures that are in the shape of tubes. For example, these tube-like structures, and vesicular structures. These vesicular structures in the shape of vesicles. These fill the cytoplasm of parietal cells when they are inactive, when they are resting. Once they are activated by any mechanism, the vesicular portion, the vesicular structures will bind with the intracellular canalicus and will make it deeper, and the tube-like structures will bind with the intracellular canalicus, producing the microvilli. And in this way, the cell will return to the active shape, to its active form. So this is a special feature of the intracellular canalicus is a special feature of parietal cells, not found any other cell in the body. So what are the differences between parietal and chief cells of the gastric epithelium? Now, we talked about most of these features. Parietal cells are found in the upper part of the gastric glands, whereas chief cells are found in the lower part. The parietal cells are larger, whereas the chief cells are smaller. The cytoplasm is acidophilic in parietal cells due to the presence of mitochondria, whereas male chief cell cytoplasm is basophilic due to the presence of rough endoplasmic reticulum. Now, regarding the vesicles, in chief cells we have secretory vesicles found in the apical part of the cells. What about parietal cells? Now, regarding the acid secretion, of course, hydrochloric acid is not stored inside the cell. And as you know, we have just the pumps that are part of tubular vesicles and that become expressed on the surface of the membrane when the cell is activated. And what about intrinsic factor, the other substance released by parietal cells. It is also thought that intrinsic factors is stored within the tubular vesicles themselves. It is synthesized as any other factor by endoplasmic reticulum and Golgi, but they are stored inside tubular vesicles, and when these vesicles adhere to the cell membrane, intrinsic factor is released to the outside by exocytosis. So these are the main differences between parietal and chief cells of the gastric epithelium. Now, parietal cells produce hydrochloric acid into the lumen of the stomach, and the pH in the lumen of the stomach is very low, less than 2. And this low pH is important for digestion, but it's also very dangerous and very harmful for the mucosa, for the cells. So how can the mucosa protect itself from the hydrochloric acid secreted into the lumen of the stomach? Several mechanisms are present for this protection. First, the mucus and bicarbonate secreted by the surface epithelium. This is the mucus, as we have said, is thick and contains a lot of bicarbonate ions, and these bicarbonate ions neutralizes the hydrochloric acid. So actually, the pH at the surface of the stomach is high. It is about neutral. Then we have the surface epithelial cells have tight intercellular junctions and ion transporters that maintain the hydrogen and bicarbonate ions concentrations. Also very important is that we have rapid turnover of the surface epithelial cells. So the time from the birth of the surface epithelial cells until their death is about 5 days. So every 5 days, new surface epithelial cells are formed. If you compare, just remember, we have just talked that the lifespan of parietal cells is 200 days. So you can see how the surface epithelial cells have very rapid turnover, whereas parietal cells have a long lifespan. Finally, we have extensive blood vessels in the lamina propria, the underlying connective tissue. This provides nutrients, remove toxic material, and help replace damaged cells. This extensive blood vessels. So these are factors that protect the gastric mucosa from the harmful effect of hydrochloric acid. Failure of these factors will make the gastric mucosa susceptible to damage by the hydrochloric acid. And this will ultimately lead to ulceration. Damage to the epithelium is called ulceration. If this will involve parietal cells, this will lead to vitamin B12 deficiency. Remember that one of the functions of parietal cells is the secretion of intrinsic factor, and this intrinsic factor is important for the absorption of vitamin B12. So if we have damage to the parietal cells, this means that we have reduction in the release of intrinsic factor, and this will lead to reduction in the absorption of vitamin B12. Now, vitamin B12 is important in the process of erythropoiesis, the formation of red blood cells. So if we have deficiency of vitamin B12, this means that we will have reduction in the formation of red blood cells, and this will lead to anemia. Anemia caused by vitamin B12 deficiency is called pernicious anemia. So ulceration of the stomach that leads to damage of parietal cells is accompanied by pernicious anemia. Finally, we have the enteroendocrine cells. From their name, these are endocrine cells. They are found in the lower part of the gland, in the lower part of the gastric glands, and of course, they secrete hormones. The secretory granules of these cells are usually found in the lower part of the cell. These are endocrine cells that secrete hormones. The hormones pass into the blood vessels. Blood vessels, where are they found? They are found in the lamina propria under the epithelium. So these cells produce their secretion into the underlying connective tissue lamina propria. This is why the secretory granules are found in the lower part of the cell. Now we have many types of enteroendocrine cells. We divide them according to the hormone they secrete. One example is G cells. G cells of the stomach secrete the hormone gastrin. There's another way to divide the enteroendocrine cells of the stomach is into closed type or open type. This is according to whether they are in contact with the lumen or not. In the closed type, here, this is the enteroendocrine cell. It rests on the basal lamina. It is resting on the basal lamina, but the other cells surround it completely so that the enteroendocrine cell is not in contact with the lumen. It is not in contact with the lumen. And this is what is called the closed type. The closed type. The open type, on the other hand, here we have a wide base resting on the basal lamina, and it is here where we find the most of the secretory granules, and also we have a thin apical process. Thin apical process. This process will pass superficially until it reaches the lumen. At the tip of the process, we have several microvilli. We have microvilli. So in this type of enteroendocrine cells, the cell is in contact with the lumen through its apical process and microvilli. Now the microvilli of these cells actually act as chemoreceptors. They detect the contents of the lumen of the stomach. So these are the chemoreceptors. They detect what is found inside the lumen of the stomach, and according to what is found, they may release different types of hormones. So this is the open type of enteroendocrine cells of the stomach. Now, the regional differences in the mucosa of the stomach. Anatomically, the stomach is divided into 4 regions: the cardia, the fundus, the body, and the pylorus. Histologically, however, the stomach is divided into 3 regions only: the cardia, the pylorus, and the fundus body. The cardia and the pylorus are small regions, whereas most of the stomach is considered in the region of the fundus body. Histologically, the cardia has simple branched spiral glands, which are mainly mucus secreting. In the pylorus, here we have deep pits. The gastric pits are deep, and 2 to 3 spiral glands open into the pit. And in the pylorus, the glands are mainly mucus and gastrin secreting. Whereas in the fundus or the body, the pit is not deep, and we have more glands opening into it, about 5 to 7 tubular glands open into each pit. All cell types are found in this region, mainly parietal and chief cells. So the main function of the stomach is performed by the glands found in the fundus of the body, because they have the majority of parietal and chief cells, whereas the cardia and the pylorus are mainly mucus secreting, and gastrin is mainly secreted in the pylorus. Now here one must think, why in the cardia and why in the pylorus the glands are mainly mucus secreting? Now, one of the main functions of the stomach is the secretion of hydrochloric acid performed by parietal cells. And as we have said, acid is very harmful, very damaging to the mucosa. But the gastric mucosa is protected against this hydrochloric acid. Now think about it. The cardia is the site where the esophagus meets the stomach, and the pylorus is the site where the stomach opens into the duodenum. Now, although the stomach is well protected against hydrochloric acid, the esophagus and the duodenum are not. So this is why in the cardia and the pylorus we have more mucus secreting, because as we have said, this mucus protects against the harmful effects of hydrochloric acid because it contains bicarbonate ions. So just in case some acid passes in this region, it is neutralized by the mucus found here in both these regions, the cardia and the pylorus. So again, in the cardia and the pylorus, we have excessive mucus secretion to neutralize the acid, protecting the esophagus and the duodenum. So far, we talked only about the epithelium of the stomach, and now we continue with the rest of the layers. The mucosa of the stomach, as we have said, the mucosa is formed of epithelium, connective tissue, lamina propria, and muscularis mucosae. The lamina propria is a loose connective tissue layer. This surrounds and supports the gastric pits and glands. It is highly vascular. This is the usual. Any connective tissue under the epithelium is highly vascular. And it also contains some smooth muscle cells and some lymphoid cells. The muscularis mucosae, this is a smooth muscle cell layer that separates the mucosa from the submucosa. So this muscularis separates the first layer of the gastric wall from the second layer, the submucosa. The submucosa, the second layer of the gastric wall, is a connective tissue layer. It is present under the mucosa. Now, here we have some lymphoid tissue, some macrophages, some mast cells. These are all play part in immunity, but importantly, we have the submucosal plexus. The submucosal plexus is a group of nerves, of course. It is part of the enteric nervous system, which controls the muscular activity and the glandular secretion of the alimentary tract. Muscularis externa, the third layer of the gastric wall. It has the three sublayers of muscle: outer, longitudinal, middle, circular, and an inner, oblique layer. So we have outer longitudinal muscle layer, a middle circular muscle layer, and an inner oblique muscle layer. Why do we have three sublayers of muscles in the muscularis externa? Because this will help to mix the food found inside the stomach with the gastric juice. And also you must remember that in addition to chemical digestion, the stomach also performs mechanical digestion. It breaks down food particles mechanically by pressure, so it requires good muscle strength. This is why we have three sublayers of muscles in the muscularis externa of the stomach. Now, the circular layer in the pylorus is thickened. This thickened circular layer in the pylorus will form the pyloric sphincter. The pyloric sphincter. Now, here it is well known and well taught that the pyloric sphincter is an anatomic sphincter. It is called anatomic sphincter because we actually have a structure that we can see, which is a thickened circular layer of muscle, thickening the circular layer of muscle in the pylorus. Whereas at the junction between the esophagus and the stomach, the cardia, we have an esophageal sphincter, but it is not anatomical. It is not anatomical because in this region we do not have thickening of muscle. We do not have thickening of muscle, neither in the esophagus nor in the stomach. This is why we do not say it is an anatomical sphincter, but we say it is physiological sphincter. Also, within these sublayers of muscles, we have the myenteric plexus, of nerves, and this is the second part of the enteric nervous system, which controls the muscular activity and glandular secretion of the alimentary tract. The outermost and the final layer of the gastric wall is the serosa. Serosa, which is the same as the visceral peritoneum, it is areolar connective tissue and mesothelium. We have a layer of areolar connective tissue covered by a layer of simple squamous epithelium, which is called mesothelium. And it is continuous, the serosa is continuous with the lesser and greater omentum. So these are the histological features, the main histological features of the different layers of the gastric wall.